Good afternoon, everybody. Welcome to Laboratory Medicine Grand Rounds. I think we'll get started. So I'm John Tate from the Department of Laboratory Medicine, and today we have an interesting presentation. I think this is a record for speakers in one session, and we'll explain the rationale for that in a little bit. But let me first just introduce the speakers. So Colin Pritchard, he received his bachelor's degree from the UW and MD, PhD degrees from the UW, and completed residency training in pathology, all at the UW. And he's been an assistant professor in laboratory medicine since 2011. Dr. Tom Walsh received his bachelor's degree from University College in Galway, Ireland, and then a PhD from the University of Leicester in the UK. And he's been on the faculty in the Division of Medical Genetics at the Department of Medicine at the UW since 2005, and is currently research associate professor. And then Dr. Mary Claire King needs no introduction, but I'll introduce her anyway. She received her bachelor's degree from Carleton College, PhD from Berkeley, was then on the faculty at UC Berkeley for many years, and we're very fortunate that she was recruited to the UW in the mid-90s, and that's when I had a chance to meet her. And she's in medical genetics and genome sciences, and I think her career has accomplished about as much of about 100 ordinary scientists. But there's one paper in particular that really stands out, which I still remember reading, and that was her 1990 paper where she identified a locus on chromosome 17 linked to hereditary breast cancer, and that turned out to be what was later called the BRCA1 gene. So that's really kind of almost the entire foundation of the field of hereditary cancer risk, and we can trace the work discussed today directly back to that really pioneering work. And finally, I'm John Tate. I received my bachelor's degree from Harvard and MD, PhD from Washington University, St. Louis, residency training here at the UW, and I'm currently professor of laboratory medicine. So now I'll turn it over to Colin Pritchard, who will introduce the talk.
Well, thank you, Dr. Tate, for that nice introduction. So the talk today is called a CPC for the 21st century. So for those of you who don't know what CPC stands for, that's Clinical Pathologic Correlation Conference. And Dr. Tate will talk a little bit more later about what that means. So here are the learning objectives. I'm not going to go through them. They're in your handout. So what we're talking about here is next-generation sequencing in the clinical lab for clinical diagnostics. And so first of all, what do we mean by next-generation sequencing? So next-generation sequencing is a series of technologies where you can do massively parallel sequencing, that you can generate orders of magnitude more data than conventional sequencing, such as Sanger-based sequencing. And so what are the theoretical advantages of this sequencing method in clinical diagnostics? So you can analyze up to a whole genome in a single run. With this deep sequencing, where you get maybe thousands of sequencing reads per base pair, you can detect rare subpopulations. So if you're talking about germline mutation testing, you might be able to detect a low-level mosaic mutation. If you're talking about sequencing tumor tissue, you may be able to detect a low-level subclone. It can detect all classes of mutations with deep sequencing. So by that I mean point mutations, small insertions and deletions, large insertions and deletions, even genomic rearrangements. And it's efficient one-step testing. So traditionally it's been quite expensive to do clinical testing of more than a single gene at a time. And with next-generation sequencing technologies, we're now for the first time able to assess multiple genes in one assay efficiently. And the other nice thing about this technology is that it drives research and discovery. So there are potential disadvantages of the next generation sequencing technology in the clinical space. So one of them is information overload. So I've heard people refer to the information tsunami. And so there's sort of two aspects to this. One is that just understanding, well, how do you make sense out of all this data? Incidental findings become very important. And the other thing is just the actual logistics of how do you store the data. We're finding that clinical labs such as our own are having to have large servers, having to come up with protocols to store very large amounts of data, et cetera. So there is a need for software and decision support. to be able to analyze this scale of data. So people that have been doing clinical sequencing for years are used to looking at Sanger sequencing traces and be able to sort of interrogate every single base with their eye, and they feel very comfortable with that. That's just not possible in this world, so we rely on software and decision support to some extent. Cost is currently still somewhat high, but it's coming down very quickly, and it's already, I think, more cost effective to do a gene panel by next generation sequencing than it is by Sanger sequencing. And I think within a couple years, these costs are going to come down to the point where it'll be very clearly less expensive to use this technology than the sort of traditional or older technologies. Turnaround time is a potential disadvantage, but again, with improvements in instrumentation and improvements in laboratory workflow, these turnaround times are coming down as well. So next generation sequencing, I really want to emphasize, can be sort of one-stop genetics shopping. So traditionally, we use different methods to detect different classes of genomic variation. So I've got a list here of types of genomic variation and then a technology that's used clinically. So for large deletions and duplications, we can use microarrays, we can use fluorescent in situ hybridization or other methods, but we can just use next generation sequencing. Rearrangements, we can do classical cytogenetics with G-banding or fluorescence in situ hybridization. Next generation sequencing detects that. Small insertions and deletions, again, there's various methods for this. Sanger sequencing will typically pick these up, but again, we can detect them with next generation sequencing, and then of course single nucleotide variants, many different methods to detect those. But again, next generation sequencing detects it. So it really is potentially one-stop shopping, although I'll emphasize that that depends on sort of what we call the depth of coverage and how deeply you sequence, and some other technical details, whether you're able to reliably detect all classes of variation with next generation sequencing. So what are some considerations for clinical next generation sequencing assay design? So I've already sort of emphasized this, that deep sequencing is needed. So, I think in the clinical space, when there's really a patient's diagnosis on the line, we want to be able to detect all classes of mutations, not just point mutations. And so deleterious mutations tend to be not point mutations. So a lot of the deleterious mutations, frame shifts and so on, tend to be insertion deletion mutations. So we really want to be able to detect those, and to do that, deep sequencing is necessary. And it would be nice to be able to detect mosaic germline mutations or tumor mutations that are in an admixture of normal cells, which again, deep sequencing is needed. So we want to minimize incidental findings. So there's a balance between wanting to sequence as much as is clinically relevant, but not more than that. And so sequencing a whole genome at this point, while technically feasible, does pose some problems for incidental findings, especially in the clinical space. So that's something we want to minimize. And so I think these considerations make current, what I'll call generic exome sequencing, exome is sequencing all the exons in the genome, or whole genome methods less suitable for clinical testing. Although I've put this sort of caveat, I think that these technologies and our knowledge are improving so rapidly that in the near future, I think exome sequencing and whole genome sequencing will be sort of ready for the clinical space. So targeted gene panels is what we're going to talk about mostly today. And so these are panels of between 10 and 40 genes where we're sequencing the whole gene. But the genome is something about 25,000 genes. So we're talking about a very small slice of the genome. And so if we do a targeted gene panel using this next generation sequencing technology, the advantages are it allows very deep sequencing of those regions. It's easier interpretation of data because we're only looking at even though we're looking at more genes than we've been accustomed to in the clinical setting, we're still looking at a relatively limited number that we know a lot about. It's definitely more cost-effective to do a gene panel, and you can do higher throughput, and you get fewer incidental findings. So what are these sort of next generation sequencing gene panels at UW? So we have 3 assays that we're going to focus on today. The first one we call ColoSeq. This is an 11-gene panel. It's focused on colon cancer risk, specifically Lynch syndrome and polyposis syndromes. We have another assay that was developed by Dr. Walsh in Dr. King's lab called BROCA and that's a 40-gene panel for cancer risk with a focus on breast and ovarian cancer. And then we have a third test that we call UW Oncoplex, which is a 194-gene panel for tumor-based testing. And so this is a test on tumor tissue, unlike ColoSeq and BROCA, which are for cancer risk and test blood. for germline heritable mutations. UW-Oncoplex is a test on tumor tissue to look for mutations that are actionable, that may have treatment implications for cancer patients, and may even direct which cancer therapy those patients get. And the UW-Oncoplex really is not intended for germline cancer risk. So these three tests are going to be illustrated in the three cases we're going to present for this clinical pathologic correlation conference. And with that, I'll turn it over to Dr. Tate.
So just to explain a little bit about the title and how we got here. So all of you who've been through medical training know about the CPC, Clinical Pathologic Conference, either from actual conferences at your own medical school or from the famous series published in the New England Journal of Medicine. So this is a case-based instructional method developed at the Harvard Medical School in the late 1800s. And it's based on actual real patient cases seen at the Massachusetts General Hospital. And those cases were published by MGH for many years. But in 1924, the New England Journal of Medicine took over the publishing of that, and they published them ever since. So it's very interesting to go back. Now they have their entire archives online, and go back and look at the very first, or one of the very first cases that was presented, which I've given the details here. And it's fascinating to see what was being done at the time. And it's surprising how much laboratory testing was being done. There were many blood glucose measurements being made. And insulin had just come into use. So that was the brand new targeted therapy of its day, which had just started to become available a year or two before that. Since the four of us who are speaking today are involved in the review and interpretation of results for these next-gen sequencing panels, and people started to ask us, is there a way that we could sit in on sign-out sessions and learn more about that? And we had so many requests, and we decided that Dr. King's office was too small to accommodate 20 or 30 or 40 people at the same time. So we came up with the idea of having something like a CPC. So we'll be presenting three actual cases, and with the idea being that as technology evolves and new diagnostic methods are introduced into pathology, that these can then become part of the instruction of real cases. So the first case, and these, as I said, are cases seen in our own system, either UWMC or SCCA or both. This was a 46-year-old man who was diagnosed with colon cancer last year, and as part of the workup during endoscopy, in addition to the cancer, there were many hundreds of polyps that were detected throughout the length of his colon. So after surgery to remove his colon, he was then seen in the GI Cancer Prevention Clinic at the SCCA to be evaluated for a possible familial polyposis syndrome. And his mother did not have cancer. She was alive and well, and his maternal family history was mostly negative for cancer, so that was not particularly informative. And the father's family history was also unrevealing, mostly because the patient had been out of contact with his father and his father's relatives for several decades. But nevertheless, given the clinical presentation with so many polyps, there was a fairly high likelihood this could be a genetically caused syndrome. So there are two things that are thought about in that situation: hereditary polyposis syndromes, either autosomal dominant familial adenomatous polyposis caused by mutations in the APC gene, or a more recently described autosomal recessive syndrome due to mutations in the MUT-YH or MyH gene. And genetically, both of those are possible in this family, given the sparse genetic history. So there wasn't any reason a priori to rule one in or out over the other. So the test that was ordered was the COLA-seq test, and that includes both the MUT-YH and APC genes. And the results were expected to influence the patient's medical management and the genetic counseling of his relatives. So I'd like to turn over to Tom.
So as Dr. Tate indicated, there is clinical overlap between Lynch and polyposis syndromes, particularly in respect to age of onset and the number of polyps that can present. So as a consequence, clinicians are faced with a dilemma of which test to order. And based on the current Sanger sequencing technology, these tests are quite expensive. So we thought it would be cost-effective and beneficial to patients to evaluate all known colon cancer predisposition genes in a single clinical assay simultaneously. So we developed ColoSeq over the course of about a year, and it currently evaluates 11 genes. The 5 genes, mismatch repair genes, that are known to be responsible for Lynch syndrome, the 2 polyposis genes, APC and MUT-YH, and then we also wanted to include genes that are responsible for more rare cancer syndromes, but can include colon cancer and polyposis among the spectrum of cancers. So amongst those 11 genes, the mutation spectrum, of course, includes small mutations that introduce premature truncations, such as point mutations and small insertion and deletions. But they also include larger genomic rearrangements, such as deletions and duplications that can either delete or duplicate an exon and then lead to truncation of the protein. CNVs, copy number variants, are currently detected by a separate, distinct assay that often measures the copy number either by quantitative PCR or an approach called MLPA. So we felt it was really important to be able to detect CNVs using our next generation sequencing reads. So we developed a read depth algorithm to identify these CNVs. And this cartoon here indicates the raw data. So here's a normal read depth output and below. And in here we have a trough of reads, which are then converted to a deletion call, indicated in red here, and a pileup of reads, which would indicate a duplication of an exon, indicated in blue here. So we spent about a year, because this is a brand new technology, we spent about a year validating it and assessed the accuracy, sensitivity, and specificity over the course of a year. And we saw 100% concordance between clinical sendouts and the ColoSeq mutations that we obtained through next generation sequencing. And this was true across the spectrum of mutations, point mutations, indels, and CNVs. And we published that data earlier this year in the Journal of Molecular Diagnostics.
Okay, so I'll talk about the data that we got on this first case and the interpretation now. So before I get into the actual data, I'll spend a little bit of time on this first case to talk about how we approach the review of this data as a group. As Dr. Tate indicated, there's been a lot of interest in how do you sign out next generation sequencing data? By sign out, I mean release a clinical report. So the sort of general way that we approach this is we come up with a short list of variants that are selected by each member of a multi-member interpretation team, all 4 of us in this case. We then combine our 4 lists that, again, are independently and blindly, we come up with these lists, and then we combine our 4 lists. We call these our combined faves, or favorite variants that we're going to look at. And then we come together, the 4 of us as a group, and look at the list that and decide as a group which ones are going to go into the report and which ones aren't, and what we're going to say about them in a report. So this is a screenshot that obviously you can't see, but this is just a very tiny snippet of what the actual raw data for sign-out looks like. So we have hundreds or even thousands of variants per patient, even when we're doing these targeted gene panels. You can imagine what this would look like if we were looking at whole exome or whole genome data. So it's about 300 to 3,000 total variants per sample that we detect. And so, of course, the vast majority of these variants are benign. We don't want to put 3,000 benign variants into a clinical report. That's not going to be useful to someone who's trying to treat their patient. So we need a strategy to filter out the benign variants and to sort of kind of separate the wheat from the chaff, and just to select the variants that we're going to look at for further consideration. So this is a sort of schema of our filters in general. So we rely very heavily on both internal and external frequency databases. So we have an internal database of, I think it's actually over 2,000 patients now, where we can look at the frequency of a variant, of how frequently we've seen it before. And then we also can look at external databases such as the Exome Variant Server, which is an excellent resource that's hosted here at the University of Washington, dbSNP. For copy number variants, there's the database of genomic variation, DGV. There's also the 1,000 Genomes Project, and then there's mutation-specific databases. So we rely on all of these to look at what is the frequency in the population. Because of course, if a variant is at 50% frequency, it's probably not causing a rare, highly penetrant syndrome. So that reasoning is relied upon. So if we do identify a clearly pathogenic variant, something that's a truncation or something that's already been reported as pathogenic in a mutation database, we're done. That's definitely going into a report, of course. The trickier thing is variants of uncertain significance, which of course when you're looking at so much data, you're going to find variants that no one's seen before, and you're going to find variants that people have seen before, but they don't really know what they mean. And so for variants of uncertain significance, we go through another sort of set of filters where we use computer tools and silico tools. Specifically, we use polyphen2, SIFT, and mutation taster to predicting whether a missense variant is more or less likely to be damaging. We also look at evolutionary conservation in several ways. And then for splice variants, we have, or for suspected splice variants, we have in silico tools to tell us whether that variant is more or less likely to have an effect on splicing. Okay, so if we get to the data on this case 1, this patient that had 100 polyps in his colon. So by coloseq, there were 639 total variants. But when we look at variants that are just less than 1% frequency in the population, we get all the way down to 20. So you can see how that filter very quickly gets us down to a relatively manageable number of variants. And then if we just focus on the variants that are in the coding exons or splice regions, variants that are more likely to be important, we get down to 4. And then if we look at these other tools that I discussed, we get down to just 2 variants in this case that are predicted to be possibly damaging. And so this is what the short list looks like for review. So this is the short list from case 1. So we usually divide it into different classes of mutations. So we have our single nucleotide variants. I'm abbreviating SNVs here. In this case, we did have a single nucleotide variant on the short list. It was an MLH1 variant. But when reviewing the literature and mutation databases, there's actually a lot of literature on this variant, and the vast majority supports that this is benign. And so the decision was made that probably this didn't need to be reported because it was very likely benign. We didn't have any indels or insertion deletions. We didn't have any small insertion deletions on our short list. But the most interesting variant was a copy number variant. So there was a APC coding exon 1 to 4 duplication that we detected using the methods that Tom outlined. And this was very likely to be pathogenic and causal of the phenotype. So this is what the data looked like. So again, using the sort of pileup of reads, what we call the normalized depth of coverage, it's very clear that we have in the blue region a duplication of this region of APC. And again, this is detected directly by sequencing. We're not looking at array CGH data here. We're looking at sequencing data and normalized depth of coverage. We have a second method we can look at, so we can actually not only look at depth of coverage and pileup, but we can also look at split reads. So because we're doing sequencing, we get some reads that actually sequence across the breakpoint. So in this case, we're talking about a duplication, and I'll show you that it's a tandem duplication. And so we get sequencing reads that actually sequence across that breakpoint, and then we can use that as corroborating evidence. And that's what this is showing here. And so this is what we're actually looking at. And so sort of there's this tandem duplication of APC coding exons 1 through 4. And then the black and red sequencing reads are basically a sequence that can't happen in a normal genome. It's sort of, it's out of order. But you can sort of do the Sudoku puzzle and figure it out, and then you can sort of reason out that this is in fact a tandem duplication. So not only, we have multiple lines of evidence, and we also did confirm this by targeted PCR as well. So we have multiple lines of evidence that strongly support that there is a APC duplication, and this is predicted to result in loss of function of one APC allele. So this is the clinical report. I'm not going to read through it, but just to emphasize, this is reported in the medical record just like any other lab test, and this is the actual report for this patient. And so the final diagnosis in case 1 is familial adenomatous polyposis due to a tandem duplication of coding exons 1 through 4 in the APC gene.
Okay, so our second case will illustrate another test, the BRCA test. And so this case was a 51-year-old female who had ductal carcinoma in situ that detected in the right breast and then shortly thereafter in the left breast, and both breasts were surgically removed. So after the surgical treatment, then she was evaluated in the genetics clinic, and her family history was strongly positive for prostate cancer on her father's side, and selected other cancers were present on both sides of the family. And I've indicated what they were. So clearly there's a lot of cancer there, but it isn't falling nicely into one of the clearly recognized classical hereditary cancer syndromes. But nevertheless, it's still worth doing some kind of genetic evaluation. So the test ordered in this case was the BRCA test, which is a panel of 40 genes that increase the risk of breast and ovarian cancer, and some other cancer types as well. And so the purpose of this then would influence both the patient's own medical management and importantly also, if a mutation was found, could provide guidance to her relatives on their cancer risks in the future.
So BRCA was the first clinical assay that we developed back in 2009. And again, it's an effort to increase the throughput and effectiveness of gene-by-gene screening, so doing all screening in one shot or one assay. So it was set up essentially for breast and ovarian cancer patients that had received wild type or negative BRCA1 and BRCA2 results from commercial sequencing. But in addition, it also is used now as a hereditary cancer syndrome test because it does include so many other known cancer genes. So at present, it sequences 40 known and emerging cancer genes, including ovarian cancer genes, RAD51C and RAD51D, and emerging cancer genes such as ATR, which seems to be responsible for oropharyngeal cancer, and BAP1, which has been reported in cases of uveal melanoma. So as I said, we have been using BRCA since 2009, and it's exceptionally well established in the clinical setting. We've now run more than close to 3,000 breast or ovarian cancer patients in the research setting. And last year, we published a study with Elizabeth Swisher, where we looked at 360 patients that had had their primary surgery at the University of Washington over a 10-year period. And a large fraction, of course, did have BRCA1 and BRCA2 mutations, but when we looked across a larger panel of genes, an additional 6% had germline deleterious mutations in any of 11 genes. So we drew the conclusion that approximately a quarter of all ovarian cancer patients do have a deleterious germline mutation in any of 13 genes.
Okay, so I'll talk about the data interpretation for case 2. So going again through our total variants, so with BRCA, we're looking at a larger number of genes than coloseq, in this case 40 genes, so it's not surprising we have more variants to start with. In this case, there were 1,649 total variants detected by BRCA. When we do our filters on frequency, looking for rare variants, we get down to 55 variants. 20 of those are in coding or splice regions. And of those, the only two that looked very interesting were 2 frame shift mutations, both of which looked to be deleterious to the protein. So this is what our short list looked like. So in this case, we didn't have any single nucleotide variants that were of interest for sign out. We had 2 small insertion deletion or indel mutations. One was a single base deletion in a gene PALB2, which is a gene that's well established as a breast cancer risk gene with about a 2 to 6-fold increased breast cancer risk when patients have inherited mutations in PALB2. So we did decide that this was important to report. The second mutation is in a less well-characterized gene called FAM175A, also known as Abraxas. Again, a deletion mutation predicted to result in a frame shift and loss of function of one allele. And this is a gene that has been reported associated with breast cancer risk. However, this particular mutation, or even this class mutation, had not before. And we didn't have any copy number variants that were of interest for further consideration. So for this case, I thought I'd show a little bit about how we actually visualize the data. So those of you who've done Sanger sequencing may be familiar with looking at the sort of red, blue, black, green traces of the bases and sort of evaluating the data. So we can't really do that with next generation sequencing, but there are tools like the Integrated Genomics Viewer that's made by the Broad Institute, which I'm abbreviating IGV here, that will allow you to sort of look at the sequencing reads sort of piled up on their gene. And so what we're looking at here is each of these gray bars is an independent sequencing read, and this is just a very small snippet. We actually have over 500 sequencing reads here, and I'm only showing you about 20 or 30 of them. And then these black lines indicate the one base pair deletion. So this is just sort of a gut check. It's nice to be able to, when you have a deleterious mutation like this, to confirm, to actually put eyes on data and really confirm that, yes, in fact, there is a deletion mutation in this case here. And so that's what we're looking at. So for our other frameshift mutation in FAM175A, we also detect it by the Integrated Genomics Viewer view. And this was particularly important, and I'd like to highlight in this case that we actually didn't have great depth of coverage. So even though on average we have hundreds or even thousands of sequencing reads per base pair, there are some regions of the genome that aren't covered as well. And so this is an exon 1 of FAM175A that's a very GC-rich region. And we only had, I think, about 20 or 30 reads covering that region. So it was still enough to detect this mutation, but had we not been doing very deep sequencing, we may have had 0 reads there. But because we had relatively low reads, we did want to confirm this by Sanger sequencing, which we did, and it did confirm, to be at fully heterozygous levels. So it's analytically correct. And so obviously you can't read this, but this is the sort of long-winded report that went into the medical record. In this case, it's a complex report because we have 2 deleterious mutations that may be additive. One in a very well-characterized gene, PALB2, that is well-characterized in its association with breast cancer risk. The second in a gene that has been reported associated with breast cancer risk but is less well-characterized. So the final diagnosis, increased breast cancer risk due to an inherited deleterious mutation in PALB2. This is C.3323-A. And then possible additional contribution of the FAM175A deleterious mutation to breast cancer risk.
So the third case that we'd like to present this afternoon is a case of a 76-year-old Asian woman, lifetime non-smoker, who had developed lung cancer several years ago and had been treated with several different treatment regimens over the last 2 years, and had recently, her disease was progressing based on growth of a lesion as detected by CT scan of her lungs. So this is a situation that commonly occurs, that people progress through the various treatment options, and at a certain point they start to run out of possibilities based on what is conventionally available according to standard classifications. So this patient had recently had molecular testing of her tumor, and I hesitate to say sort of standard, because this wasn't standard until just a couple of years ago, and we have the 2 people here that I think introduced the test. We've got Colin Pritchard, and I believe Susie Dinsis is here as well. So those will detect mutations that predict response to certain targeted therapies, but she was negative for that. So the physicians treating her decided to order the UW Oncoplex test, because that takes a much broader view of the potential somatic mutations present that could be useful to provide new therapeutic choices. So the purpose of this then was to provide either new therapeutic options or possibly to guide eligibility for clinical trials either here or elsewhere based on her molecular test results.
So as Dr. Tate said, this assay, which we developed over the course of this year, is designed to identify specifically actionable alterations in tumors to guide therapy. And a well-known example of an actionable mutation is BRAF V600E in melanoma, where there is a targeted anti-BRAF therapy called Zelboraf that seems to decrease size of tumors in the patients with these malignant melanomas. We did have to alter our protocols in the laboratory to make this work for lower input DNA samples. And of course, most of the clinical specimens in this case are formalin-fixed paraffin-embedded tissue, so it did require some additional validation. The UW Oncoplex version 2 currently sequences exons from 194 genes. It's tiered here into currently actionable genes such as BRAF and EGFR. Tier 2 is genes that will be actionable in the near future, such as the MAP kinases, many of which are currently in clinical trials. And then genes which are frequently mutated in various tumor types, such as P53. Oncoplex does differ from the commercially available panels in that it assesses all exons, not just the known hotspots of these genes. So we validated on a large series of tumor samples over the course of this year, all of which had had prior testing in laboratory medicine. The samples were from lung, colon, melanoma, and AML. As Colin said, we do get very high coverage for this assay. It's a lot smaller than the germline panels, so the coverage is almost always over 1,000x. And this enables detection of tumors in a background of normal or stromal cells. We also detect gains and losses of genes with an algorithm developed by Steve Salaponte. And again, using split reads, we can detect certain translocations and inversions, such as the ALK EML4 of inversion on chromosome 2 in lung cancer.
Okay, so I'll talk about data interpretation for case 3. So I'm just going to jump right in with our copy number variation output to show you kind of what this looks like in a tumor. So when we're looking at a cancer, all bets are off in terms of copy numbers. I mean, tumors can have wild cytogenetics, as those of you who look at this can know. So this tumor had many gains and losses. But the key here is we want to focus on things that are actionable, that are going to be helpful for a cancer patient at this time, that have some implication for therapy. Most of these, I mean, maybe 10 years from now we'll know what to do with them, but we don't. So the two that we've kind of focused on here is there's a gain in the MYC gene, looks like MYC amplification, which is a very frequent event in many cancers, including lung cancer in this case, with over a 4-fold copy gain, even in the setting of admixture with normal cells. And then there's a loss of the RB1 gene, which is a very frequently lost gene in many cancers. This is a cell cycle regulator, one of the sort of classic tumor suppressor genes. So we flagged these for our short list. In addition to those, we didn't have any single nucleotide variants on our short list, which is, again, I'd like to emphasize, you know, technologies that are only looking at single nucleotide variants are often going to be missing, you know, the meat of where these mutations, important mutations are. So we didn't have any that we flagged on this that we thought were actionable. We did have 2 insertion deletion mutations, small insertion deletions. And the first of which I think is the most important. So ERB2 is also known as HER2 or HER2-neu. And there was an in-frame insertion in exon 20 of ERB2, which is actually very well characterized, activating mutation, and very well characterized in lung cancer. And there's data in lung cancer patients about specific responses to treatment. And so the good news is that it actually predicts sensitivity to certain tyrosine kinase inhibitors. So we were very excited about this. There was another loss of function mutation in a gene called ETV6. This gene was in the tier 3 and not clearly clinically actionable at this point. So even though this was analytically real, we don't really know what this means for the tumor, so it's not as critical for this patient's care. As I showed in that earlier slide, there were copy number variations. There were many of them. The 2 that we sort of flagged was MYC amplification and RB1 loss. And so in searching the literature, there actually is some data that suggests that there's possible implications for tyrosine kinase inhibitor therapy in lung cancers that have MYC amplification. So we decided that that was worthy of discussion. RB1 loss, you could argue there could be some effect on cell cycle, but there wasn't any clearly actionable data in lung cancer at this point that we could find. So in our clinical report, we reported the ERB2 or HER2 exon 20 insertion and the MYC amplification, and we suggested treatment with second-generation tyrosine kinase inhibitors that target not only EGFR, the epidermal growth factor receptor, but also ERB2, which is in the EGFR family, because there is some data in lung cancer patients that they will potentially respond to those tyrosine kinase inhibitors, but not the traditional ones that are used like erlotinib and gefitinib. And in fact, they may even be more resistant to erlotinib and gefitinib. And so this patient was actually currently on erlotinib, so this had double implications. One, maybe this patient really wasn't on the right therapy. This mutation predicts that her tumor would be resistant to that therapy. And two, there's another therapy that may be indicated. And this patient actually did get on to lapatinib, which is a second-generation TKI. So it's very early on. We don't know whether this patient's going to respond yet or not, but we're very excited and hopeful that the patient will. So the final diagnosis in this case, lung cancer with ERB2 exon 20 insertion mutation, suggests treatment option of a tyrosine kinase inhibitor that has anti-ERB2 activity, such as lapatinib or afatinib, and there are others.
Thanks. It falls on me to summarize the cases that we've seen and to introduce some interesting points for discussion. First, I would like to emphasize that in the Seattle way of things, the gentlemen, none of them, have bragged about the fact that all three of the products that we have talked about were made locally. They were. Broca, Coloseq, and Oncoplex are all created by these guys. This is not some fancy-dancy Eastern imported technology. This is our guys. Right. I want to tell you a little bit about the current capabilities and complexities of using these three tools. When Tom first developed Broca in our lab in 2009, as soon as next-generation sequencing was operationalized, as soon as we had our first platform for next-generation sequencing, we were very interested in how to make this approach available to patients. We were inspired to this by Robin Bennett, by Gail Jarvik, by Liz Swisher, and we, of course, Tom and I, as PhD scientists, were very involved in research on patients and their families and have worked with thousands of families and have given them, I think, very useful information, which they have shared with their clinicians. But our clinician friends said, look, we need to make this usable in the clinic for patients that come in every day, not only for patients in the research setting. Fortunately, Colin Pritchard and John Tate shared that ambition, and what you've just heard is the tip of a very large iceberg of what has happened since. So, what do we detect? What do we detect that is not yet useful to you as clinicians? And what are the challenges of so much information, much of it novel? So first, what do we detect with this targeted genomic capture approach, what Tom calls a one-stop shop? Given current technology, it's our conviction that targeted genomic capture is more efficient for the clinician than either whole exome or whole genome. All of those are perfectly feasible technologically, but as you've seen, the balance of subtle mutations, the range of mutation types, the range of genes that can be hit, really motivates very deep sequencing for a targeted number of genes and genomic regions so that one can return information quickly and have very great coverage for all mutations, inherited or somatic, of all classes in all targeted genes and loci. These are presently all actionable variants that are involved in cancer development, whether or not they've ever been seen before. You've heard about point mutations. You've heard about frame shifts. You should be aware that much of the software that is available generally does not detect frame shifts efficiently. This technology does. And you've heard about genomic duplications and deletions, which are also not universally efficiently detected. Note also that many patients, including at least one of the ones you've heard discussed today, who have inherited mutations in one of the critical breast or ovarian or HNPCC genes do not have a strong family history of the relevant cancer, so would not have been sent for expensive, slow, commercial, conventional sequencing of each of a large number of genes. But their attending physician said, let's give this a shot with one of these approaches that detects a vast number of mutations, all mutations in a vast number of genes, and they were successful. Second, what do we detect that's not yet useful to you? First, there are a very large number of rare mutations, the great majority of them missenses, that is, amino acid substitutions, that have clinical significances that are not yet known. As you might anticipate, this is an area of very active research in cancer genetics. It pertains both to inherited mutations and to somatic tumor-based mutations. We do not report these back to you unless there is experimental evidence that the mutation actually alters the gene in which it occurs. We do not report back to you a variant simply because it's there. There has to be some experimental evidence, typically a functional assay, as for CHEK2 or PALB2, that the mutation actually alters the gene. Truncating mutations, of course, alter their genes. But these missense mutations fall on a spectrum from completely benign to absolutely devastating and everything in between. So we, at this moment, are reporting back, as a general rule, only devastating mutations, which means only a fraction of the missenses. This is a moving target. Next year at this time, we will be reporting back a larger number of missenses because there will be functional assays. A second category of information we detect that is not yet useful to you, it comes from a non-scientific source, and that is the complexity that's introduced by patent protection on BRCA1 and BRCA2. As you may know, the challenge to the exclusive enforcement of patent protection on BRCA1 and BRCA2 is presently being returned back to the Supreme Court. So in the next year or so, this will be resolved one way or the other. At present, until it is resolved, we establish a firewall between our research use of these tools, which of course detects BRCA1 and BRCA2 along with everything else, and our clinical use of these tools that is covered by patient insurance. What is covered by patient insurance does not include BRCA1 and BRCA2 because of the patent issues. Our research activities, which are in the same lab, run on the same platform with the same tools, of course, do include BRCA1 and BRCA2. These are different activities. Research activities, no money changes hands. Clinical activities, of course, the insurance of the patient is charged for the test. And third, what are the challenges of having so much information, much of it novel? As you've heard with the three examples that were presented by my colleagues, a vast number of variants appears for each patient for each test. We parse this information for you. We do not return to you 639 or 1,503 variants for a particular patient. You're not caught in the weeds working with this information. For me, one of the most pleasant things about this entire experience has been to exchange information with John Tate, Tom Walsh, Colin Pritchard, about each variant in each gene that we see. Each of us has a real depth, decades worth of knowledge about a subset of these genes, and put together, we know a lot. And we also, of course, have friends, many of them in this audience now, that we call on when we have a particular variant in a particular gene that we don't know personally. So that kind of expert knowledge applied to each patient, which is, of course, the way of life around here every day, is particularly useful in this kind of setting. We're all learning a lot. The four of us are committed to continuing this together forever, so that there will always be this kind of expertise brought to bear on this extraordinarily large range of genetic variation that we see for each patient. Having said that, the amount of relevant information we return to you is still substantial. And what does that mean for you as a clinician? The first thing that I think it will mean is that genotype sometimes trumps phenotype. That is, you may well learn of a mutation, inherited or tumor-specific, in your patient that is a known mutation. It's known to be devastating, but you hadn't previously ever seen it associated with that phenotype. You'd seen it associated with a different cancer, but not this cancer. For example, there might be a mutation that appears in an ovarian cancer that is unquestionably a deleterious mutation and is known to be a driving mutation, say, in lung cancer. And we're now seeing it in ovary for the first time. We send you that information. These are your patients, and you need to decide what to do. There may very well be a treatment based on that. You need to decide how you want to proceed. The same thing can, of course, occur in the inherited context. Finally, from the point of view of genetics, every patient that you see who carries an inherited mutation represents a family. So for every proband, every patient with an inherited mutation, it will be important to make the information known to the family so that as yet unaffected relatives can be tested and followed if they have high genetic risk. That is, every patient represents a family. This is, of course, a strength here because we have a very strong medical genetics clinic that can collaborate in the identification, validation of mutations in family members and counseling of these families. This is why God made medical geneticists, and they are the perfect complement to the information that we are giving to you. I'll close simply by noting that if population screening were introduced using these three tools, which I think will happen over the next fairly short period of time, we would prevent a great many cancers. Suppose, for example, that we made available to every woman at the age of 35 BRCA testing, and suppose also that patent issues didn't obtain. We would prevent 1 in 4 ovarian cancers. So that's where this is headed. Right now, this is available whenever you, as the physicians of patients that come to your attention, believe they are needed. And we hope that it will be of service to you over the next many years. Thanks very much.
Thanks, Mary Claire, for that excellent summary. We're very lucky to have Dr. King at the University of Washington, and I feel very fortunate to be able to collaborate with her and Tom Walsh, and also my mentor, Dr. Tate. I'd also like to acknowledge, first of all, I'd just like to acknowledge there are a huge number of people that have done very important work building up to being able to do this kind of clinical testing. So many excellent physicians, genetic counselors, medical technologists, and other healthcare providers who've contributed to the care of patients whose cases were presented today in particular. So we weren't able to sort of acknowledge everybody associated with the care of those 3 patients, but we're very grateful. And also the many volunteers for their contributions over the years to the research that has made this testing possible. So, the groundbreaking research that Dr. King has done and others, have really led up to this being possible. So this is the official acknowledgments. So we have many, many people to acknowledge, and I realize it's getting late on time. But I'd like to just draw attention to a few people in our 2 groups. So in lab medicine, Christy Smith, Karen Kohler, Steve Salaponte, and now Tatiana and Sheena are all very important members of the team, as is everyone else on this list. Our colleagues in pathology have been integral to this, particularly Susie Dinsis and Raj Schmidt, Yanjuan Liu. And then the King Lab, of course, is an equal partner in this. In her group, Ming Lee, and bioinformatics, Liz Swisher, and Ann Thornton, who's also been very helpful to our technologists, as has Tom, in really kind of holding our hands and teaching us this technology, and the many other people. And also our colleagues in medical genetics, particularly Robin Bennett, Gail Jarvik, and others, and colleagues at SCCA, particularly Renata Martins, Keith Eaton, Carrie Stricker. So does it take a village to do next generation sequencing? I'd say the answer is probably yes. This is just to sort of illustrate the sort of large number of people who have been involved in this effort, and we have an amazing team, and I feel very fortunate to be able to be working in this area. So thank you very much.